I'm going to be presenting on behalf of our UO1 project that's studying aging in the human hippocampus. So the prevailing model for how memories are stored is through specific neuronal communications, and that can be formed by specific synaptic connections, as well as the strength of these signals through the synapses, such as fine-tuning the amount of neurotransmitter release. But as we age, there's known memory impairment that is contributing to the known age-related cognitive decline. And the hippocampus region specifically is critical for memory consolidation, as well as other functions that are known to have age-related decline, such as learning, spatial navigation, and emotional regulation, making this region highly studied when it comes to cognitive decline with age and age-related memory loss. And it's also known to experience pretty strong physiological changes, such as age-related atrophy. While there have been transcriptomic changes that have been described in the aging brain, and most consistently, these have been related to a decline in genes involved in synaptic function and an increase in genes that are related to the immune system, contributing to chronic neuroinflammation. However, the regulatory mechanisms that are underlying these transcriptomic changes with age are not fully elucidated. So to understand the mechanisms related to these age-related transcriptomic changes in the aging brain, we profiled um, features of gene regulation, such as how the 3D genome chromatin folding changes with age and finding activity of putative enhancers in their target genes through aging. So through epigenome profiling, we can identify these gene regulatory programs that might be underlying these transcriptomic changes with age. However, as we all know, the epigenome is quite diverse from cell type to cell type, as one genome can give rise to several epigenomes. So there's a critical need to resolve these cell type epigenomes, and that can be achieved through single cell profiling. So to achieve this, we employed two different single nucleus multiomic profiling assays. One is the commercial 10x genomics multiome to get single nucleus attack and single nucleus RNA-seq. And the other is the SNM3C-seq assay to achieve single nucleus DNA methylation and 3D chromatin. This assay, SNM3C, was developed in Joe Ecker's lab, and a variant of it in Bing Ren's lab at UCSD. So we use these assays across hippocampal tissue from 40 donors that spanned adulthood, that are balanced between each 20-year age group between 20 to 100 years old, with sex balance of 5 donors in each age group that are female and 5 donors that are male. And so to study the gene regulatory programs that change with age, we use these assays to generate cell type resolved maps, the transcriptome, chromatin accessible landscapes, DNA methylomes, and 3D chromatin organization. And this is a collaboration between Bing Ren's group, specifically Dr. Soyan Lee and grad student Sainath Mamdi, and with Xiaomin Chu's group at UC Irvine. And so we profiled around 300,000 nuclei using the 10x multiome and around 22,000 nuclei using SNM3C-seq, giving us 4 molecular modalities in each major subclass in the human hippocampus. And this allowed us to reconstruct gene regulatory programs in each cell type, where we can make predictions for putative enhancers in their target genes by integrating these modalities using the activity by contact, or ABC model, giving us cell type specific putative enhancers in their target genes that show cell type correlated activities. So to identify what features are changing with age, we could correlate different features such as gene expression across the donors to find out which significantly correlate either in the negative or positive direction across the donors with age. And this is just showing an example of this gene CACNA1A that has a negative expression correlation with age in astrocytes. And so we quantified features such as genes, chromatin accessible candidate cis regulatory elements, and differentially methylated regions in each cell type. Notably, we see a big decrease in genes in astrocytes with age, and we find microglia in particular have very age dynamic epigenomes, both for chromatin accessibility and DNA methylation. And when we look at the dynamics of change for these age-correlated genes, we see similar dynamics in each of the cell types where the expression changes start to occur in donors around age 50, and then there's a pretty striking change after that. And so there's most of the gene regulatory changes occur around that middle age group. And these genes are enriched for different functions in different cell types. For example, we see immune genes highly activated in the microglia and loss of ATP synthesis genes in several neuronal types in astrocytes. And we notice a very striking proportional change in astrocytes mostly, where with age we see a very marked decrease in the number of astrocytes. And we captured this in both our 10x multiome and SNM3C assay. And we validated this using immunostaining for an astrocyte marker and found that this can be replicated in the immunostaining of the CA3 region of the hippocampus. So we wanted to understand why these astrocytes could be declining with age. And this could be related to age-correlated synaptic dysfunction, as astrocytes have a very critical role in maintaining proper synaptic signaling through recycling of neurotransmitters and removing excess glutamate in the synaptic cleft to prevent aberrant synaptic transmission from neighboring synapses, and also release of gliotransmitters back to the presynaptic and postsynaptic neurons. So when we look at the gene regulatory programs that change most strongly in astrocytes, it's overwhelmingly enriched for genes involved in ATP synthesis, so genes involved in the electron transport chain, as well as ATP synthase subunits, which decline in ATP synthesis could lead to programmed cell death as well as non-apoptotic cell death, such as autophagy. And we do see genes upregulated in astrocytes with age related to autophagy. But what controls these genes that are changing expression with age? When we did motif enrichment analysis on the peaks that change with age, we find very strong enrichment for the NRF1 motif at sites that have lost accessibility with age. And consistent with that, we see a gain in DNA methylation at NRF1 motifs. And this was nice to see because NRF1 is actually known to be an activator for genes involved in oxidative phosphorylation and ATP synthesis. So this suggests that NRF1 activity changes with age is leading to mitochondrial dysfunction and potentially the loss of astrocytes in the hippocampus with age. And as I mentioned, we see microglia have strong activation of immune response genes, and this has been replicated from many studies. So we wanted to understand or try to figure out which transcription factors might be activating these genes with age. So we scored every TF motif for changes in accessibility and DNA methylation with age. And we see a group of TF motifs that really stand out. These are all BZIP family transcription factors that have a strong gain in accessibility and a corresponding decrease in DNA methylation with age. And their predicted target genes are activated with age in microglia, suggesting that these are responsible for these age-related changes in the transcriptome in microglia. And we see very strong, in addition to chromatin accessibility, very strong changes in DNA methylation in the microglia with age. So we wanted to investigate what these corresponded to. And to our surprise, they corresponded to two major cell states in the microglia, as opposed to gradual changes for methylation at these sites. These corresponded to two kind of stable states that we called micro 1 and micro 2, and these are age associated. As you see, the younger donors, the 20 to 40-year-olds, are mostly contributing to the micro 2 cluster, and this shifts with age to where the 80 to 100-year-olds are almost fully the micro 2 cell state. And these are associated with DNA methylation changes near genes related to development, where with age, the developmental genes, that most of these are expressed in early development. They gain methylation with age. And the micro 2 cluster has hypomethylation near genes involved in immune responses such as chemokine production, suggesting that these methylome reprogramming events could be establishing a more inflammatory microglia with age. And when you look at the dynamics of the methylation across the donors, so these are the DMRs, or differentially methylated regions, that go down with age or up with age, you can see it really just corresponds to the proportion of these micro 1 and micro 2 states across the donors, where you see that up until around 50 years old, all the donors are predominantly micro 1, but then you start to see this switch happening to the micro 2 state in donors between the ages of 50 to 75, where above that you see almost all micro 2 microglia existing in these aged donors. And so because we have this dynamic age group where the proportions of these states don't correlate with age, we can disconnect changes with age with changes with the switching of this state. And we find that there's distinct TF motif enrichment for the age correlated versus the primed state TFs. So we see ETS family enriched for the correlated CCREs for the state, for the switching, while age correlated TFs are enriched for these hormonal responsive TFs, such as glucocorticoid receptors and androgen receptor TFs. So to try to understand what this state could represent, we looked at genes that are upregulated in the aged micro 2 state, and we see overwhelming enrichment for the antigen presentation complex, the MHC class 2 complex, is dominating the gene ontology enrichment. So when we look to see what this could represent, there's a literature on what's known as the primed microglia cell state, which is described in rodents, both in mouse and rat studies, where microglia can be immune primed through some sort of priming stimulus. This can be aging. This could also be other sort of immune insults, such as infections or surgery or TBI, which cause this transient immune response, which is then shut off through expression of anti-inflammatory cytokine expression. However, upon another immune insult, there's a heightened immune response, giving them this immune memory, giving them this designation that they're in a primed state. However, it is not known how this primed immune memory is encoded. So our data is actually suggesting that is encoded, at least in humans, through a DNA methylome reprogramming, a relatively stable epigenetic mark that allows these microglia cells to quickly and have this exaggerated inflammatory immune response upon secondary immune insults. And although we didn't find this clearly through our RNA and chromatin accessibility clustering using an unbiased clustering, we can find this same state in ATAC-seq single-cell data if we use supervised clustering using these methylation features that correspond to these two states and looking at chromatin accessible sites at these regions. And this can separate out age-related clusters in both our study and also other published single nucleus ATAC-seq from different groups, where we can find even in different brain region this correspondence between the micro 1 and micro 2 with age. And next, we wanted to look at 3D genome reprogramming in these epi-primed microglia, and we identified these contacts that get stronger or get weaker with between the epi-primed and the micro 2 state. And we find that the contacts that get stronger in the primed microglia are enriched for type 2 interferon response genes that become activated with age. For example, this gene IL-15, a pro-inflammatory cytokine that's greatly activated with age. We find this contact forming between the promoter of IL-15 and this upstream putative enhancer that gains accessibility and loses DNA methylation with age, suggesting that there are these widespread 3D genome reprogramming events in combination with the DNA methylation reprogramming events, establishing this immune-primed microglia in the aging hippocampus. Okay, and lastly, we noticed that there's this general erosion of the homeostatic 3D genome organization in the most aged donors. So I'm showing two cell types here, microglia and oligodendrocytes. And you can see there's a very strong gain in trans chromosomal interactions in these 80 to 100-year-old donors in both cell types. And that corresponds with a decrease in the contacts within topologically associating domains, also in the 80 to 100 donors in these same cell types. So we see this general erosion of the homeostatic 3D genome organization in the aged donors across multiple cell types. And we wanted to understand what could be causing this loss of the homeostatic 3D genome. And we know that CTCF is a very important factor in 3D genome architecture. So we perform CTCF cut and tag in young, these green violin plots here in aged donors, and we see this global loss of CTCF binding through cut and tag. And I'm showing an example locus here with these different CTCF binding sites. You can see a dramatic loss in the CTCF cut and tag signal. And to try to understand if this is related to the loss of the TAD structures with age, we looked at chromatin accessibility at CTCF motifs in each cell type to predict CTCF binding at cell type resolution. And we find that the degree of CTCF decline of accessibility in each cell type correlates with the cell type's strength of the TAD contacts. So really implicating that the loss of CTCF with age is resulting in this decay of TAD structures. And to summarize, we see a dramatic loss of astrocytes in the aging human hippocampus. We see that microglia undergo this age-associated switch to an epigenetically primed cell state, which is stably encoded through the DNA methylome and 3D genome reprogramming. And we see that these 3D genome restructuring is corresponding at sites where we have activated CCREs and activated interferon gamma genes. And we see this global erosion of the homeostatic genome architecture, which is corresponding with a reduction of CTCF binding. And this is a preprint currently on bioarchive, so feel free to explore that. And we also, with our collaborators at WashU, at Ting Wang's group, they helped us establish this data hub where you can explore this data on the WashU Epigenome browser. So I'd like to acknowledge those who contributed at UC San Diego, Dr. Xiaomin Shu's group at UC Irvine, and our collaborators at WashU. And in the remaining time, I'm happy to take any questions. Thank you.